Hi, my name is Safia Khurshid. I am an assistant professor in Stanford Research, Sioux Falls, South Dakota, and my lab studies alternative splicing of RNA in pediatric cancer. Previously, we were not able to use the publicly available data sets and ask splicing questions because I did not have a full-time bioinformatician or a data scientist working for me. But now, since the past a few months, I've used Claude Code and tapped into many of these data sets and basically gotten very transformative, incredibly transformative results. And so for this project, we've used one of these data sets, which is from TCGA, which is a liver cancer data set. It has RNA sequencing information, it has the proteome information, and it also has the survival of the patients linked with it. And so we ask this question, whether the alternative splicing is correlated with the survival of these patients. So we got a lot of splicing variants, and 382 of them did significantly correlate with the overall survival. We then wanted to make our analysis more stringent, so we used two independent pipelines rather than trusting one supercomputed splicing table. So we recodified using these two independent pipelines, and they both agreed on 90 genes, which we ran again for survival, and we were able to find 18 genes that were significantly correlating with survival. So then one of the important things we found was that we were able to go back and look at the gene expression data very quickly of these tumors, and we found that 8 out of these 18 genes, the survival was associated only or solely with the splicing change in these tumors and not with their expression. And that result, I think, is not really well known in the field or in the drug discovery field. So we were able to so now I think we're able to show how important the splicing of these genes is, and we can now have the ability to validate it either in my wet lab or also in multiple different cohorts, which I think is transformative. So the important thing that came out was that this was a work that could be done with the help of bioinformaticians and data scientists, but could take weeks, if not months. But this way, with Claude Science, I was able to do it in days, and I think we're now able to ask obviously bigger questions, bolder questions, and transform how we do research. Thank you for this opportunity.